Asexual reproduction is the formation of a new individual from a single parent without the fusion of gametes. Thus, in asexual reproduction, the genetic constitution of the offspring is exactly similar to that of the parent. Asexual reproduction is also known as agamogenesis, as it does involve meiosis and the fusion of gametes. Different groups of plants show different modes of asexual reproduction. In algae such as Chlamydomonas, reproduction takes place by formation of motile flagellated zoospores within the parental body. Zoospores, when released, develop and form individual organisms. Algae such as Eulothrix also show formation of motile zoospores that divide mitotically to form new organisms. The fragmentation seen in Spirogyra is another mode of asexual reproduction. In Spirogyra, accidental breaking of the filament leads to the formation of new individuals that survive independently. Fragmentation is also seen in bryophytes such as moss, where the branching protonema may develop into new plants. Let's look at the different modes of asexual reproduction in gymnosperms and angiosperms in more detail. The generation of a new plant from the vegetative parts of a plant, such as the root, stem, and leaf, is called vegetative propagation or vegetative cloning. Vegetative reproduction is seen in herbaceous perennial plants that show structural modifications of the stem, roots, and leaves. Modified underground stems that act as vegetative structures include tubers such as potatoes, bulbs such as onion and garlic, corms such as colocasia, and rhizomes such as ginger. Did you know that the beautiful tulip flower also propagates itself through underground bulbs? Subaerial modifications also help in the vegetative propagation of plants. A runner is a slender branch that rises from the base of the stem and runs along the surface of the ground. Plants such as the strawberry are propagated through runners. The stolon is a modified stem which arises from the base of the stem. It rises and then bends down to form a new plant. Mint and jasmine are plants that have stolons. Another example of a modified stem is the offset. It is a short shoot that arises from an axillary bud at the base of a stem and gives rise to a daughter plant at its apex. Water lettuce and water hyacinth reproduce through offset. Water hyacinth propagates itself so rapidly that it causes depletion of oxygen in water, thus causing the death of fishes and other aquatic organisms in the pond where it grows. It is thus called blue devil or Bengal terror in India. Another modified stem, the sucker, shows a lateral branch which arises from the underground region of the stem, travels a short distance underground, and then grows upwards to form a new plant. The banana plant is propagated asexually by the formation of suckers. Plants that propagate themselves through modified roots are sweet potato, asparagus, and cassava. Some plants such as the bryophyllum propagate themselves through leaves. The leaves show the presence of small buds at their notches. The buds fall off and grow into new plants. Some plants belonging to genus Lilium produce small bulbs in their leaf axils or in place of flowers. These are known as bulbils. The bulbils, when shed from the parent plant, grow into new individual plants. In plants such as wild garlic, the bulbils grow on flower heads and sprout to form new plants. In flowering plants, the fusion of the male and the female gametes leads to the formation of the zygote, which divides mitotically to form the embryo. However, there are some plants that produce seeds without fertilization. Formation of seeds through asexual reproduction is called apomixis, also referred to as agamospermy. In some plant species, the embryo develops directly from the diploid sporophytic cells of the nucellus or the integument of the ovule. In this case, the embryo sac or the female gametophyte does not take part in the formation of the embryo. In citrus plants, the nucellus cells start dividing and enter the embryo sac to form many embryos. This occurrence of more than one embryo in a seed is called polyembryony. Horticulturalists have used vegetative propagation for the cultivation of many commercially important plant species. Vegetative propagation ensures that the desirable qualities of the plant are maintained from one generation to the other, and that plants are produced in large numbers with minimum inputs. Artificial methods of vegetative propagation include cutting, grafting, layering, and tissue culture. In cutting, a vegetative part of the plant is cut and rooted to develop into a new plant. Examples of plants propagated by stem cuttings are sugarcane, rose, blackberries, pineapple, potato, and China rose. In grafting, a stem cutting of one plant, scion, is inserted into the stem of another rooted plant. Soon the two stems unite and the inserted cutting grows from the rooted stem. This brings the characteristics of two or more plants together. Apple, peach, walnut, pear, and avocado are examples of plants that are propagated by grafting. In layering, the lower branch of a plant is bent downwards and covered with moist soil. This branch then grows roots and a new plant develops. The garden strawberry is propagated with the help of its runners. The modern biotechnological technique of culturing isolated plant or animal cells or small pieces of tissues and organs under controlled conditions in a sterile growth medium to obtain clones of the plant is known as tissue culture. In the plant tissue culture technique, a vegetative part of the plant body is excised and cultured in a suitable culture medium to obtain many miniature plantlets. Tissue culture technique has been successful in increasing plant yield, obtaining virus-free strains of plants and obtaining hybrid plants such as the pomato. However, hybrid seeds must be produced every year to prevent segregation of desired character. This is done by producing apomictic seeds that the farmers can store and use every